Hi, and welcome back to the HeFPipe tutorial. In this video, I'll touch on some important points regarding the input files we discussed in the previous video. So let's dive right in. Regarding allele reports, I already mentioned briefly in the previous video that the first allele report in the allele report directory, when sorted alphabetically, must contain every sample used in all of the other allele report files. So let's take a look at what that looks like. So here's the first allele report in the Allele Reports directory in the example data. And we can see that it does in fact contain every sample in my data set. So this is easy enough to do if you know that one of your allele reports contains all of the samples you use. Just rename this file so that it's the first in alphabetical order in this directory. But let's say that none of your allele reports contain the entire set of samples. Well, in that case, you will need to make a dummy allele report, and this dummy allele report would contain every sample and each sample's corresponding genotype at a dummy locus, right? So these genotypes would be totally fake, and that dummy locus would then just be added to the loci to exclude file, which in the example dataset you'll remember is called monolist, right here. So you're just adding a fake locus to the monolist, and then you're seeding genotypes for that fake locus for all of your samples, and you're making that a dummy CSV file, allele report. And you have to make sure that the dummy allele report file has a name that would ensure that it's the first file in alphabetical order in this directory. So I've actually included an example data set with a dummy allele report so that you can see how it works. And it's in the directory called 3 with dummy allele report, which is right here. And here's the dummy allele report example. So it looks exactly like, oops, that's not it, that's it. The other samples, right? It has sample names for all of the samples, and then it has a totally fake locus with totally fake genotypes. Okay, the next point I wanted to make is quick and kind of a corollary to the point about making the first allele report all-encompassing. All the other allele report files must contain at least a subset of the samples that will eventually be included in the analyses. So none of these other files, besides the first one, can have samples that aren't in this first file. Next, there are several quick points to be made about the samples, locus, and trait names, most of which are common sense, but are built into the assumptions that the HeFPipe pipeline is making. The first one is don't name the same samples differently across different allele reports. So if there is a sample that's called 1 here, it can't be called 3 in another allele report because these things are going to get combined. 2 is blanks that you don't want to include and are not in the rejected sample list have to contain the word blank, any permutation of caps, in them. So an example of that would be here. So remember that if we put any of these numbers, right, 1, 2, 3, 4, 5, 6, 7, 8, whatever, in the rejected samples list, it gets excluded from the relevant analyses in the pipeline. But in addition to that, actually blanks get excluded no matter what. So if any of your sample names have the word blank in them, they get excluded. So only call things that you want excluded blank, and don't include the word blank in anything else. The next thing is that samples should be named according to the following convention. You have to include a number and then other information, which is optional. So in other words, designate each sample with a number, and if the sample names must contain other information for some other purpose in your analysis, that's fine, but the pipeline will ignore all symbols including and beyond the first non-digit character. So this means that while the sample numbers must be consistent across allele reports, so 642 can't be called some other number in an allele report, the other information is allowed to be different. Right, so this sample could be 642 and then some other completely different character string in a different file, as long as it started with 642 and that was the last digit. The next point is don't name any of your loci any permutation of the word pop. And the reason for this is that when you do the gene pop formatting part of the pipeline, it actually uses the word pop when formatting. So don't name any of your loci with the word pop. And the same is true for your population name. Don't name that something pop. Okay, next point is that locus names cannot contain the dash symbol, and because the symbol will then be used as a subtraction operator when the analyses are passed to Piper. So don't put dashes in your locus names. So the next point is that locus names and trait names cannot contain spaces. So if necessary, just replace all the spaces with underscores in the allele reports, in keep list, in monolist, and or in the acceptor file if we're talking about fitness traits. So an example of this would be in this allele report jam session, the space gets replaced with an underscore. And you can see Cripple Creek, that's not true, but Cripple Creek is actually on monolist, so it gets excluded from analyses anyway, so it doesn't seem to matter with the spaces with that. But just to sort of cover your butt, no spaces in any of the locus names and no spaces in any of the trait names. So what I mean by trait names is if you go to your trait containing file, which again is called acceptor.csv in the example data, none of these headers have any spaces in them. Okay. Similarly, the next point, don't name any of your traits or loci with any permutation of the word done. And the reason for this is that it will interfere with the regression and single locus association analyses in the pipeline. So none of these headers can have the word done in them. Next point is your population name, which you'll be prompted to enter right before the pipeline conducts the gene pop format conversion, cannot have the same name as any of your loci. So don't call any of your loci the same thing that you're going to call your population, and vice versa. And finally, there are two last, more general points that I want to make about HeFPipe. The first one is that heterozygosity-fitness associations are generally not appropriate to conduct on multiple populations. So HeFPipe is accordingly not equipped to accommodate multi-population data. And the second thing is that the current version of HeFPipe is only configured to analyze diploid data. So make sure that you have no loci in any of your allele reports that contain only one column of scores. So none of your genotypes can be haploid genotypes. And that's it for this video.